Good morning, everyone. Thank you for giving me the opportunity to give this talk. So now that we know very well all the advantages of using zebrafish as a model, I'll be happy to present you the fast deletion mutant zebrafish model to study ALS that our team has previously created, and now I'm continuing these studies. So I will briefly introduce the ALS Fast Pathology. Fast is a major gene associated with the ALS pathogenesis and cause some of the most severe cases of ALS because it's indeed characterized by a young age onset and a rapid disease. Indeed, 35% of the familial ALS cases occur to under 40 years old. Fast encodes for protein that is part of RNA binding proteins. That means that it's implicated in several roles of the RNA metabolism, including its splicing, the trafficking, and translation. Fast is an RNA DNA binding protein. It's ubiquitously expressed in many cell types, but in the CNS, it's mostly expressed in the nucleus. So the majority of the Fast mutations are localized in the exon 15, that encodes for the NLS, the nuclear localization signal, and mutations on this provoke the redistribution of Fast into the cytoplasm. So mostly focus their work on the possible nuclear import of Fast, but it's known that how Fast mutations exert their pathogenicity can occur by two main mechanisms. Again, a function pathway that indeed is the accumulation of the protein in the cytoplasm, and these in form of stress granules and may be toxic, or a loss of function pathway that by the nuclear import of Fast into the cytoplasm can happen that by missing its presence in the nucleus, this can be detrimental for the transcriptome and the metabolism. So what our lab was supporting, it's a loss of function mechanism, and it's what they basically created 1 year ago. And we were able to basically generate and to do a phenotypic characterization of this zebrafish fast deletion mutant line. What we basically do, as we explained also before, after obtaining the model, we could see that the loss of function of Fast impacted the lifespan by decreasing it. Indeed, starting from day 6, more than 80% of the homozygous embryos die. And also we could see that this phenotype was persisting also at day 3, 4, and 5 as we determined it by this automated behavior protocol. So loss of function on Fast, we could see that by the touch-evoked escape response, how they were swimming. And we can see that for the Fast homozygous embryos, this is characterized by an early fatigue and almost paralyzed behavior. And we also tested the ability of the human Fast gene to rescue this phenotype. And this was possible, as we can see from the increase in terms of distance and velocity of the swimming for the homozygous embryos. And what they also saw that together with these behavioral disabilities, we could see also some anatomical defects, including shorter motor neuron axons and also fragmented neuromuscular junction that they could see by basically the presynaptic and postsynaptic elements failed to colocalize in the case of the Fast homozygous. And also, just for the moment in a qualitative level, we could see by marking actin filament with the fluorescent phalloidine that there is a disorganized pattern in the muscle fibers for the Fast homozygous individuals. And by marking with a more specific antibody for slow muscle fibers, the muscles, we can see here that they are more curvy and not parallel muscle fibers. And this needs to be further investigated. So when I went and I checked more carefully, basically the phenotype, I could observe that in between my population of fast homozygous embryos, we had a milder phenotype when they were swimming faster, but with short distance, and a strong phenotype, as I told before, basically characterized by paralysis. And this gave us way to try to understand also at a metabolomic level why these two phenotypes are behaving a bit different. And this is what we did. So we performed our metabolomic analysis in collaboration with the platform, the metabolomic platform of Necker, and I could see that actually there is a different profile in between the fast, mild, and strong phenotype, and they also cluster together, contrary to the, this is the wild type, and the hetero and the wild type. So, thanks to enrichment analysis, I could observe that, so enrichment analysis first give me the possibility to check the 25 top pathways where metabolites are significantly deregulated. So what was interesting was that in the top of this, I could see many mitochondrial pathways involved. And knowing from previous studies in our lab that we were already focusing a bit on the mitochondria, I tried to better understand that. So in particular, I was interested to the carnitine synthesis pathway, and I found out that the carnitine it's very important for the transport of the long-chain fatty acids into the mitochondria. After these long fatty acids, they go to the beta oxidation process. That after 4 reactions, basically, they provide yes, shorter fatty acid chains, and especially at every reaction we have acetyl-CoAs molecules that after enters in the TCA cycle in the mitochondria and in the electron chain. So this it's clearly show that carnitine plays an essential role for the energy production, and I saw that this has been also reported, and it's relevant in ALS, including so basically actions like reducing the oxidative stress and also protecting motor neurons, and apparently also helping the survival of neurons and preserving the neuromuscular junction. And here it's an overview of the two phenotypes in comparison with the hetero and the wild types. So other two pathways that I observed where almost half of the metabolites were involved and were significantly deregulated were the transfer of these acetyl groups into the mitochondria and the mitochondrial electron transport chain. So again, this might be basically telling us that there is a disturbed energy metabolism in this model. I could see that I had NADH that was higher for the strong phenotype against NAD+, and maybe this can mean that NADH is not able to reoxidate afterwards, and the fact that also the acetyl-CoA were higher in the strong phenotype, it means that maybe they are not being recycled. So somehow I think that these mitochondria maybe are starving, and I'm really curious to know what's going on. So I also find this clinical trial that actually hasn't after been replicated, but they used the AcetylLCarnitine in a combination or not with the riluzole, and they could see that the patients were well tolerating the combination, and they had a higher scale on the ALS rate. And in parallel with these mitochondrial networks, we know that it's the mitochondrial morphology. It's a good indicator to know a bit more about these organelles. So we had the possibility to inject it to this post-fertilization, a mitotracker probe, that basically accumulates in active mitochondria, and by analyzing this picture with IMARIS, for the moment, again, at qualitative level, we can see that they seems to be more fragmented on the homozygous larvae. So this mitochondrial network needs, of course, to be further investigated. And also, we checked the production of ROS by injecting another molecule that becomes fluorescent when it's in contact with ROS. So from the picture, it seems to be more fluorescent, the muscles in the FAS homozygous individuals. So right now, I'm actually comparing the metabolomics results on 110 significant metabolites in between wild type and the two phenotypes of the FAS homo, and with more than 2,000 proteins, again, wild type versus FAS homozygous, with the hope to find basically some markers that can help us to better understand. And in parallel, and what we will do is also to cross-compare our omics analysis with data that are already available from our collaboration with Alberto Catanese from Ulm University that has data on IPS and biopsies from ALS patients. And it's ongoing also a drug screening targeting oxidative stress and maybe mitophagy ongoing. So thank you very much for your attention, and thank you to all my colleagues and to the organizers.
Questions? Everyone seems hungry. Let's look.
Out of curiosity, it seems that your heterozygous is not just like the wild type, but higher than the wild type. Do you have
So I think that this is all autoregulation of the FAS heterozygous, and something that the FAS homozygous can't do. So I've been actually wondering why this mild phenotype seems to be very, very close to my hetero. So I'm very interested to know basically if they were able, even if they are a FAS knockout model, to kind of compensate somehow. But I think that for my hetero, it's all autoregulation of the FAS.
I also have a comment on your observation of two phenotypes. It's very interesting. I don't know if you saw the paper by Grassano and Chiao. Very recently, they identified that there are two populations of FAS patients, those that we already knew, which were the faster progressing, and a large population, probably 1/3, which are actually much slower. So maybe this is relevant that you have two phenotypes also. You can just...
Okay, I didn't know, so thank you, and I will check. But I really think that these are actually more close to the patient situation, probably. So